so this is going to be now the pre-lab on protista, the next in the line of Unit 2. So for protista, protista is the first eukaryotic kingdom that we're going to go into. Again, there is domain archaea, kingdom archaea. There's domain bacteria, kingdom bacteria. Prokaryote was the cyanobacteria lab that was just completed. Now, for under domain eukarya, now we're going to have several kingdoms. We're going to have kingdom protista, fungi, viridiplantae, and animalia. Viridiplantae is the next unit entirely. Animalia will be the last two units for both the ones that don't have a backbone versus ones that have a backbone. So it's separated into lower and higher animals. All right, so protista. Protista, as you saw from the notes, it's kind of a mess. Why? Because it's more or less a dumping ground for eukaryotes. Basically, if you're not a fungus, if you're not a plant, if you're not an animal, and if you're not prokaryotic, you're dumped in protista, and the idea is, oh, we'll work it out later. So because it is the way that it's set up, there's no way in heck that it can be monophyletic. It has to be paraphyletic. So where you're getting underneath the kingdom, you're getting several complete stories, but not under the kingdom the whole story, so it can't be monophyletic. So you basically, it becomes like a hodgepodge almost. Do the best they can. Now understand, when I was taught this years ago, there were about 14 phyla underneath there. Then it was decided, well, that's too complicated, so let's slim it down to 7. When they slimmed it down to 7, however, there's going to be 60 cell lineages that don't fit into any of the 7 categories which were made, so what do? You make more categories, which makes it even more confusing. So by trying to do the right thing, they ended up making it worse, which happens a lot in science, and especially in classification. All right, so the 7 major phyla that are going to be found underneath protista. The most primitive one is Metamonida. Meta means, middle, meaning the middle ground between prokaryote and eukaryote. Metamonidans are going to basically, their major trait is they're going to lack mitochondria. Mitochondria, as you know, is the powerhouse of the cell, and therefore, to be a eukaryote, you should have it. Now, these individuals either never have a mitochondrion, or as they mature, they kick it out. Well, how do they get energy? Well, basically how most things on planet Earth get energy with prokaryotes is glycolysis. So it's not a whole lot of energy that they can retain. Next group moving up then is going to be the euglenoids, euglenozoa. Now, under euglenozoa, you're going to have the true euglenids, and then you're going to have trypanosomes. Trypanosomes. Now, with that, understand we're now officially then really into eukaryote, where they're all going to, from this point on, they're all going to have mitochondria. They're all going to have new bells and whistles that they didn't have before in prokaryote. They're going to have things like spindle fibers. They're going to have centrioles. They're going to have centrosomes holding their DNA together. They're going to have histones that's going to wrap their DNA around it. If they don't, that becomes a unique case. Now, from most of these individuals, understand, should have the ability to do sexual and asexual reproduction. Usually, asexual reproduction is done whenever the environment is great and healthy. Sexual reproduction is usually only done whenever you're placed under environmental duress. Because think, with the environment's going great, it just makes sense to make carbon copies of yourself, asexual. With the environment goes badly, well, making the same copy of yourself isn't going to help. You need to have change, and therefore you resort to sexual reproduction. Now, if you can't do sexual reproduction, then that becomes more of a defining trait, because you should be able to, specifically under environmental duress. Now, for euglenoids then, you're going to have 2 categories. One's going to be parasitic underneath the phyla, and then you're going to have ones which are unique, called the true euglenoids, which have both plant and animal traits at the same time, which is a very unique thing in life. The next major group is actually a superphylum, which means that it's one category above phylum and it has several phyla underneath it. So it's kind of like an umbrella term. That's going to be called alveolata. Alveolata means you have a series of small little membranes, kind of like a Golgi complex. Stacks of membranes, like stacks of pancakes, but it's right underneath the cell membrane. Underneath there is going to be 3 phyla. You're going to have Dinoflagellata, also known as Pyrophyta. Dinoflagellata means, terrible flagellates. Pyrophyta means fire algae. Why? Because some of them, meaning 20 in the group roughly, have the ability to create red tide. Well, yeah, that makes it terrible, but it's kind of like, a couple bad apples spoiling the whole, the bushel type of thing. Next then, underneath then alveolata, we have Dinoflagellata. The next category is going to be Apicomplexa. Apicomplexa makes an apical complex, and that's how it's going to use its alveoli to invade into host cells, so they're parasitic. Things that cause like plasmodium that causes malaria, leishmaniasis, or not leishmaniasis. Let's see, not Chagas. I can't think right now. I'm going too fast. Toxoplasma gondii. That's the one that causes pregnant women to shut their immune system down if they clean litter boxes, if the cat's infected. That's one of those. Now, last category is probably the most common category that you guys are used to, which would be ciliophora, which means you have small little bristles on your surface called cilia, which you can beat. Now, when we classify protista, again, the vast majority of them are going to be unicellular. However, another way to categorize them would be, well, how do you make a living? Are you autotrophic, photosynthetic? Are you heterotrophic? If you're heterotrophic, what do you eat? Or are you a disease-causing agent? One major category that we look at is going to be, can you move? And if you can move, how? Do you move with cilia? Do you move with flagella? Do you move with pseudopods? If you move with pseudopods, do you make these large lumbering arms? That's called lobate. If you make these small little spikes, that's an axopod. If you make these little filamentous ones that then link together with mucus, so it kind of looks like a fishing cast net, that's then going to be phelopods. So how do you make them? All right, so that's another category. Another thing you look at when you classify is outside your cell membrane. Do you create anything outside of it, yes or no? Do you make a cell wall? If you make a cell wall, what's it made of? Is it made of cellulose? If you have an extracellular matrix, in other words, almost like a hard covering called a test around you, is it made of silicon dioxide? Glass? Is it made of calcium carbonate? Chalk? What is there? All those are ways to classify. Other thing we look at for classification in protista is, do you have the ability to make a cyst? Cyst is similar to when we talked about in prokaryote of an escape capsule. However, cysts are made in protists for one of two reasons. One reason is the classic way, which means when the environment goes badly, yeah, you have an escape capsule, a cyst. Those would be things like amoeboid type creatures. However, you do have other ones which actually make the cysts for disease transmission. That would be going into plasmodium, the apicomplexan I mentioned earlier for yellow fever. So those three, ciliophora, apicomplexa, and then pyrophyta, aka also dinoflagellata, fall under the auspice of alveolata. Next major category underneath protista is going to be another superphylum called Straminopila, which means that on your cilia you have these fine little hairs. Straminopila is going to have everything from brown algae to golden brown algae to what we would call, what used to be the very primitive fungi, meaning the water molds, things like that. So underneath that, we're going to have the biggest category is going to be called Phaeophyta, which is the brown algae. Then we're going to have Chrysophyta, which is the golden brown algae. And then underneath that, then you're going to have Oomycodons, which are going to be the molds. Basically, if you don't clean out your shower, don't clean out your toilet, that pink film on the inside, that's those guys. If you wash your clothes and put them in the dryer, leave them in the washer, and you remember 2 days later, you open it up and you smell that funk, that's that category, okay. So, then that's Straminopila. Now we come into the categories of true algae, which then we have the red algae, Rhodophyta, and then we have green algae, Chlorophyta. Chlorophytans, as you see in the notes, are kind of a problem because they're not sure where to put them. So when I teach them, I have to teach them twice because some of the community wants to keep them in protists. Other part of the community wants to put them back into plants. Why? Because they have chlorophyll and chloroplast, so therefore they're not sure what category to put them in. The reds and the brown algae, again, when I was taught this years ago, they were considered primitive seaweed-type plants. Reds and browns, genetically, then, when they look at them, are more similar to protists, so they shove them back in that category. So the only one still up for debate is Chlorophyta. They're not sure where to put it. Last major phylum is going to be called Choanoflagellidea. Choano means collar with a flagellum. So it kind of looks like a vase or a spittoon where you have this protein sac with this contracting collar around it, and then you have this creature in it that has a flagellum that sticks out and it wiggles it. Well, how does it move? Obviously with the flagellum. How does it feed? Basically, it makes a cyclonic effect, which then brings food down into it so it can eat. Now, those are the 7 major categories, 7 major phyla, or superphyla. Then we have the big group of 60 of them, which is other. I'm not going to go into all the other ones specifically for lab, but then we have, the amoeba-type creatures. Those would be under an auspice called Sarcomastogophoran. You're going to have what are called the true amoebans, which are called Rhizopus. And then you're also going to have what are a very unique group, which are going to be called Radiolera. Radiolera is going to have a silica test. Kind of looks like a very ornate glass Christmas ornament with these large pores in it, and that's the one that would have axopods coming out of it. Then you have another group, not in the group of amoeboids, but now a new group called foraminiferan. These are the ones that are going to make the phelopods, not the axopods, and therefore they have a test, a shell made of calcium carbonate. If you've ever seen the White Cliffs of Dover off of the coast of England, those are billions and billions of foraminiferans, was sea bottom at one time. Last thing that I go over with you then for this chapter would be the lovely group called slime molds. Again, slime molds used to be put into the category of fungi, but genetically they're kind of their own category. So under slime molds, you have cellular and plasmodial slime molds. The one I'm going to show you is plasmodial, if I remember right. Okay, so for the pre-lab, that covers the major kingdoms, the major phyla we're going to go on underneath in protist. So in your lab, when you go through it, I'm just going to go with my manual and go through how I have it because I already had it set up for you. All right, so the first group you're going to be looking at is going to be Metamonida. Now, off to the side of the picture, I have a little box, and those are the features that I want you to label on the drawing, okay? So please make sure you find all of these. All right, so Giardia. Giardia intestinalis, understand, is an intestinal parasite. How would you know if you were infected with the Giardia? Basically, whenever you would defecate, there would be kind of like a slimy sheath around your feces, giving you a red flag that you have Giardia intestinalis. If not treated, you would die basically of exposure, which means that you would basically have uncontrolled diarrhea until eventually dehydrate. Now, so when you look at Giardia, understand what you're looking at is essentially, it's a poop smear because you're looking at the creatures in someone's fecal remnants that, were infected. Now, when you look at it, it looks like a bunch of shrapnel everywhere. Well, yeah, that's what diarrhea is. It's mostly water. It's pieces of your large intestinal wall, kind of like your skin flaking off, that looks like almost like tissue paper or toilet paper floating in a commode type of thing. However, what you will see in it, though, Giardia, characteristically, when it's viable, has a characteristic teardrop shape to it. So it kind of tapers. If you see that, no brainer. It's Giardia if you see a teardrop. Now, Giardia is under a group called diplomonad. Diplo means two, meaning it has two nuclei. Looks like two eyeballs looking at you, but they're not eyeballs, they're two nuclei. It also is going to have two flagella, which come out of the top like this. So therefore, when they swim, they're not swimming like a classic, when you think of a sperm flagellum that wiggles like a dog's tail, think of it like a brushstroke, and that's how they get around town. All right? That would be Giardia. Next one you have is Trichomonas vaginalis. Trichomonas falls under the group Parabasalidae. Parabasalids are unique in that they're able to take their cell membranes and push them out with their microfilaments, and almost they undulate through the water. Kind of think of like a magic carpet almost. Beautiful to look at underneath, with an electron microscope. So you have these undulating membranes. Bad to have because, as you can tell, Trichomonas vaginalis, it's an STD. It's a pathogen. So for Trichomonas, what you would see would be, again, it's a vaginal smear of a woman that was infected. So when you see it, you'll see like these pink cells, which look nice and pretty and healthy. Well, those are regular epithelial cells found in the vaginal orifice. They're normal. However, what you will see around them, it almost looks like a feeding frenzy of these other cells, which look kind of hairy, that are around it. That's the Trichomonas. What are they doing? They're feeding off of it. Okay? So basically, some of these you can have one cell and it's surrounded by like 8 Trichomonas. It's kind of like an old Bugs Bunny cartoon where, the piranha, they rip it apart type of thing. So they're especially in bad cases. All right. So that would be the two in Metamonida. Next, we go into euglenozoans. So we have true euglenoids. Now, euglenoids, understand, if you were in a lab and saw them, they literally look like little pieces of rice because the magnification isn't great. But the one I picked for you is actually, pretty zoomed in. So euglenoids kind of look like a piece of rice with a butt. So you have this piece of rice with a butt. All right? Out of the butt, you're going to have a flagellum, which is going to help you for propulsion. Another flagellum is going to emerge, come back, and go back down in. Why? It acts like a rudder for steering. Okay? You're going to have a nucleus. You're going to have a covering outside of your cell membrane. Now, that covering is not a cell wall. It's called a pellicle. Pellicle. Basically, what is it? It's a coat of protein around it, which kind of acts like molecular Kevlar. Gives you protection and flexibility. Also in this, what you normally see in euglena when they're happy and healthy, tons of green little balls floating throughout it. Those are chloroplasts. okay? Because this thing is going to be both photosynthetic and autotrophic at the same time, both plant and animal trees at the same time. Problem: euglenoids live in freshwater ecosystems. If you followed anything from Gen Bio, they're basically with osmotic pressure. It's a hypotonic environment. Water's moving in at a faster rate than it leaves. They could explode. So what do they have to alleviate that? When you see little clear areas in the cell, the little clear areas in the cell, both anterior and posterior, those are called contractile vacuoles. What do they act like? A sump pump. As the water comes in, it gets pumped back out, so therefore you don't go boom, all right? Last but not least, with the euglenoids, when they're alive, there's going to be a small little area somewhere in the cell which is going to have pigment in it. That is often called the, quote, eye. It isn't the eye, it's called the stigma, S-T-I-G-M-A. It is photosensitive, which makes sense. If this thing can swim in a freshwater ecosystem and it's photosynthetic, it would make sense. I swim toward the source of light, so therefore I can photosynthesize. Now, it's not an eye in the true sense. Basically, it can tell between light and dark. It can't see color like your eye. It can't see motion. It can't see detail. Basically, it just sees light versus dark. All right, that's a euglena. Next, we have trypanosoma. Trypanosoma is in a group called kinetoplastids. Kinetoplastids are where, then, what I mentioned earlier, where you have trypanosoma, Chagas, leishmaniasis, they're pathogens. So in trypanosoma, you're going to see a blood smear. So in that blood smear, what you would see are a bunch of little balls everywhere. What are those? Those are healthy red blood cells, erythrocytes, okay? In between them, then, you would see these little tiny, little weird mustache wormy things in between. That's the trypanosome. The one you're looking at specifically causes African sleeping sickness because, as you understand, red blood cells move oxygen around the body. If you destroy those cells, you cannot move oxygen, and therefore you cannot make ATP, and therefore shows fatigue. The bad things about trypanosomes is on their glycoprotein coat. They can change their, they can manipulate the sugars on them. So basically, when you use an antibiotic on them, say once, and 3 months later, it becomes ineffectual because they modify the outside. So usually when you see people that have trypanosoma, they're very, very sick, they get an antibiotic, then they look, oh, they're recovering, they're going to get better, and then maybe a month or two later it resurges. That's because when you give them the antibiotic, you might have killed 95% of them, but the 5% that weren't affected because of genetic modifications, they slowly build up the number, and then they come back again for another surge. These two do have two flagella, one anterior, one posterior. One's for swimming, one's for piercing. All right. Now we move into alveolata. Alveolata, again, is the supercategory, the superphylum. Underneath that, then, we are going to have dinoflagellata. Dinoflagellata, the terrible flagellates. There's all different, when you see the slide of it, it's mixed. All different shapes and sizes. One of the most common ones, think of, it's called serration. Think of it like, you remember old Star Trek from the 60s, the little emblem? It's going to look out like that. All right, so basically we have a cell that kind of looks like this. But understand when you get it in focus, it doesn't have two prongs. It's a tripod. There are three. Okay? Around the middle, there's going to be a little groove around the middle. That's where one flagellum goes around the middle like a belt. Again, for steering. You're going to have one flagellum coming out of the back for propulsion. This covering on it is going to be made up of cellulose, like you would have in a plant. But the cellulose isn't like the cell wall of a plant. It kind of, what you get is cellulose plates. If you can think of like armor, like chain maul, or if you can think of like shingles on a roof. So basically, they always look fuzzy because you have these cellulose plates and they overlap each other, giving this fuzzy appearance over the whole thing. Again, it gives you strength with flexibility. There is going to be a nucleus in this thing. They are bioluminescent. There are other categories that make them unique, which you'll see in the PowerPoint, but I don't want to belabor that anymore because you can read it as well as I can. All right, that's going to be one of the more common dinoflagellates you would see in a mixed dinoflagellate. Next one is going to be Plasmodium. Plasmodium is not a dinoflagellate. Now we're at apicomplexid, the parasitic types of alveolatans. In this case now, it's going to be another blood smear. Looks like this. And if you notice from the PowerPoint, I go through the life cycle of Plasmodium, malaria. You should know the life cycle. So when the female mosquito sticks her proboscis into you, she injects into you what is called the sporozoite. Remember, spores can be used for, or the cyst can be used for transfer. So the cyst is then transferred into your bloodstream. The sporozoite would then travel in your bloodstream till it gets to your liver and becomes the merozoite. Mero means essentially middle infectious stage. That's the one that you would see in a blood smear. That's the one that would attack the blood cells, would attack the liver. One of the hallmark signs of malaria is you get yellow fever. Why? Red blood cells go to your spleen to die. When they die, they release a chemical called bilirubin. It has a yellow tint. That bilirubin chemical would then pass through the liver, and the liver is supposed to basically filter that out. Well, if the liver is being attacked and not working very well as a filter, what happens? Bilirubin is not processed, and then you build up the yellow pigment in your bloodstream, giving you yellow skin, meaning also the palms of your hands, soles of your feet turn yellow. When it becomes almost terminal, that's when the conjunctiva, the white part of your eye, turns yellow. That's pretty much signaling, liver shutdown. However, the ones that I showed for you then are going to be the merozoite stage. Now follow. Look at it from the perspective of the infection. It's inside of you, and the environment's starting to go bad, meaning your immune system's starting to kick in and starting to try to kill it and get rid of it. So what does it have to do to get out of Dodge? It has to resort to sexual reproduction. It's then going to make gametocytes. Those gametocytes, then a new mosquito comes, it drinks your blood, and it will draw in the gametocytes from your bloodstream. The gametocytes will then fertilize within the gut of the new mosquito, go to the sporozoite stage, and go into the salivary glands of that mosquito. So when it goes to get blood from a new individual, it'll inject the sporozoite, and the cycle continues. Now again, the one you're going to see is going to be the merozoite stage. You'll see red blood cells. If they don't have anything in them, they're uninfected. If they have these little dots in them, or if they have like a little squirmy, wormy thing in them, they're infected. That means they're infected with Plasmodium. That's the merozoite stage. All right. Next, we then go into the big category people think of under alveoli, the ciliofferents, ciliates. The ones I gave for you are the paramecium, which you should have saw in Gen Bio, as well as stentor, which is a cousin of it. All right. So paramecium. Paramecium, now understand perspective here. The euglena I drew for you before, I made pretty big. If I had a euglena on the same slide as a paramecium, the euglena would look like about that big. Paramecium's about that big. So understand that paramecium, when you see it, that is the lion, that is the tiger of that drop of water. Nobody messes with this thing. It's fast and it's voracious, okay? It's going to be covered in cilia, which is why when you try to get it in focus, you can't, because again, it looks like a fuzzy jelly bean you would see on the bottom of a movie theater. Nobody wants to touch. One of those things, okay? Again, kind of like with the euglena, I'm going to have a pellicle that surrounds me for protection. Also, along with that, I'm going to have two nuclei. I'm going to have a macronucleus and a micronucleus. The macronucleus is what we call the true nucleus. It's where you would store your genes for life. How do I metabolize? How do I move? How do I do anything? The micronucleus is only going to be used during sexual reproduction. It's very analogous to what you would see in gametogenesis in basically how females make eggs. You start with one progenitor cell, it'll divide twice to make four. Of the four, one is kept, and that's what's going to be the egg. Very similar in this case, but again, it doesn't happen. This is the beginning stages of that, understand? It's not an egg. All right, along with that, I'm going to have contractile vacuoles, just like euglena, so I don't go boom in fresh water. Along with this now, let's go toward the inside. Where it makes this little indent, this is going to be called the oral groove. Basically, you would think of it almost like analogous to your mouth. In the oral groove, you'd have this little pocket right there. That little pocket is called the gullet, which is analogous to your throat. Now, there's cilia all around here, so when you find food, it wants to move it into the oral groove. Well, there's cilia in the oral groove, but it's much smaller and much tighter. Think of it like the bristles of like a horse brush. They're hard. Well, why? Because they're analogous to the teeth. So when the food comes in, these bristles will move back and forth, ripping apart the contents, and therefore making smaller food particles. It's essentially chewing its food before it puts it in the gullet. Off of the gullet, then, you'll see these little balls coming off. What are those? Those are food vacuoles. Basically the equivalent of, let's say, you chew something and you swallow it. That's how it works. This would then go to a nearby lysosome for digestion and then dispersal. Other features that this creature is going to have that you don't see in anything else prior to this. This is the first time ever we're going to have an individual that has basically an immune system of sorts, the crudest part of an immune system. Self-defense. It's going to have trichocysts, which act like buckshot, which are shot off to destroy anything in the nearby area. And it's also going to have alpha particles, which disrupt cell membranes, anything in the area. People go, well, how the hell can it release alpha particles to break apart cell membranes? and not hurt itself. Remember, it has a pellicle around it. It has Kevlar around it, and basically other things in the area, their membranes would dissociate. That's your paramecium. All right, next, stentor. Stentor is a cousin. Again, these things are three-dimensional, depending upon how they are on the slide, how they lay down, depends upon how they're going to look. Stentor, in general, kind of looks like a cheerleader's megaphone is what it looks like. So basically, you have this cone-shaped looking thing. That's going to be the pellicle. In this case now, we're going to have rows of cilia which run parallel to each other. So when you see it, it almost looks like racing stripes running down all around this cone. So therefore, it can turn, and it can also propel itself. There's also going to be cilia found around, this is the oral groove, the mouth essentially of it. The creature lives on the inside. Okay? So as these beat around and around, it makes a vortex bringing food down into it. When you look inside of it, you'll see the cell membrane right there, and then you'll see these large balls on the inside. What are those? The food vacuoles, same thing you would have seen in the paramecium. All right, so from there then, we now go to Straminapela. Straminapela, if you remember, has the brown algae, the golden brown algae, and then the water molds. Okay, so first one you see there. Now understand, from here we're going to go from unicellular to full-blown multicellular in this case. So for Straminapela, you see the next two you're going to see are going to be called Fucus and Laminaria. Fucus is a brown algae. Laminaria is a brown algae called kelp on the west coast. Fucus you have growing over here. Now, Fucus, whenever you look at it, imagine looking at like a frond, a leaf, and we're going to cut it. That's essentially what we're looking at, okay? So what you'll see, let me do Laminaria first. People usually can relate to that more. There's a rock. You're going to have this suction cup that attaches to you. That's called a holdfast. And there's a small little stem-like structure called a stipe. Now understand, the kelp can be up to 80 meters long. We're looking at 250 feet long. That's only going to be like maybe the first 6 inches to a foot, basically. Then you're going to have this massive photosynthetic blade that goes all the way up. That's the rest of this thing. That's the photosynthetic area. So the slide I gave you is I took this blade, I took the picture of the blade, and we cut it like that and look at it, okay? So for Laminaria, what you would see kind of looks like that, because you would cut the blade. What you would then see is a layer of cells on the outside. Now understand, that's not a cell wall now. These are layer tissues of tissue layers. That's the epithelium, the outside layer. And in that epithelium, you'd see individual cells. Okay, great. Those are epithelial cells. But then when you look at the epithelial cells, you'll see these strange colors, like little nodules in them. That's the pigment that makes them brown. That's called fucoxanthin, hence why they're called brown algae. Okay? Now, that's going to be the photosynthetic layer. Then when you look in the middle of it, it kind of looks like, dead cells like chicken wire. Well, what is that? That is the precursor to the beginning of what's called xylem, which in more advanced plants moves water from the root system up to the leaves of the plant. In here, there is no root system. It's just basically more or less diffusion area to move water back and forth in this big, enormous creature. The creature has one problem. It's huge. So how do I move things from point A to point B? I need a way to do that. So you have these water-conducting tissues to do that. Okay? Now, that would be Laminaria. Now, Fucus, understand, is going to take this and we're going to modify it slightly. When we get to the algae, you're going to go through something called alternation of generations. Alternation of generations means the organism is going to go through two parts of a life cycle called gametophyte and sporophyte. Gametophyte means you're going to be the haploid part of your existence, meaning N. When you go diploid, that's called sporophyte in English. Look at it a much more advanced system, okay? An apple tree. When you're looking at an apple tree, what part of it would have the full set of DNA? The roots, the branches, the trunk, the leaves. That all has the full set. Most of it is sporophyte, the 2N. What's going to be gametophyte? The apple blossom and the apple. So notice in that case, the gametophyte is tiny compared to the huge size of the sporophyte. When we're at this level, they're batting more gametophyte than sporophyte. There's going to be a term in evolution when we get to plants where then sporophyte eventually dominates. Once sporophyte dominates, that's when plants can be absolutely enormous, like a giant redwood tree. All right, so for Fucus. Fucus, again, same idea, but now we're going to modify this and put little nodules going around like that. Okay, this is the blade of Fucus. Okay, the whole thing. What you would see then is a layer around the outside. What is that? Same thing I said in fucus. That's the photosynthetic layer. You'll see the fucoxanthin in here, which will be the pigment in there. Fine. What will you see in the middle? Water conducting cells. Fine. That's all sporophyte. 2N. Full set of DNA. These little nodules are the gametophyte generation. Haploid. And in some cases, when these nodules become ripe, you can tell male from female. When you see these look like little tiny seeds or eggs all the way in, that's the female counterpart. When you see these little wispy cells toward where the opening is, that's the male counterpart. Okay? Usually when we talk about that, usually we go to archegonium is called the female, anthridium is called the male. That's fucus. All right, diatoms. So now, those would both be two examples of phaeophyta, brown algae, because again, they use fucoxanthin. Now we go to chrysophyta, golden brown algae, again, under the group Stramenopila. Chrysophyta is going to be cynora, which is a cousin of a diatom. Now, diatoms are like dinoflagellates. They can be all shapes and sizes. Most of them look like petri dishes. Think of it, okay? So a petri dish you think is round when you look at it. Turn it to the side, and it's actually a dish where you have a lid and a base. Right. The top is called the tegum, the bottom is called a scutum, and they fit together. What's it made up of? That's all made up of silicon dioxide. That's the test. The organism, the algal cell, lives on the inside. And it's going to use fucoxanthin, brown algae. Fine. Why is it called golden brown? Well, think. It's a photosynthetic creature. It has glass surrounding it. It's too dense. It's in the water. It's going to start to sink. If I sink too deep, I can't photosynthesize. How do I alleviate that? You put globules of oil on the inside of you, the golden part, and that makes you bob up in the water. So now you're golden brown. Make sense? All right, so therefore they're the golden brown algae, but that's a diatom. So there's your tegum, there's your scutum, and then the whole thing is called a test. However, understand you live in a glass house. You have to bring resources in and get rid of waste. What you'll see on these are small, what look like little air conditioning vents that are basically cut into the glass. These little air conditioning vents, excuse me, just went brain fart again because I had a cough. It'll come back to me. Damn it, I hate when I do that. Anyway, but those are the vents that let things in and out. If I think about it, I'm going to fixate, not remember it. Scoots come into mind, but that's the back of an alligator or back of a turtle. It'll come to me. I'll leave the picture up and eventually I'll come back to it. Hopefully it'll pop back into my brain. It's on your list. All right, cynora. Cynora, no, but understand, diatoms can be everything from diamond shape to rods, you name it, all different shapes. Some look like harmonicas. It's just a matter of what you look at. Almost had it and I lost it again. All right. I hate when that happens. All right, cynora. Cynora, you take the same idea, and now we're going to take these photosynthetic cells and make them teardrops, okay? However, these are colonial. So when they're in teardrops, they're going to have this clear case around them. That clear case is not a cell wall. That is the silicon dioxide test. But now you have these photosynthetic cells, and they all come to a point in the middle. So it kind of looks like, you know, a flower almost, but you have individual cells coming to a common point, making this colony up. The clear area around is called a test. Anyway, all right, so moving on then, we go to oomycota, which are basically those water molds. The one you're going to look at is called saprolegnia. Saprolegnia is the one where if you don't dry your clothes and they don't dry properly, you wash your clothes until they don't dry, that's that funk in your clothes. So this is the one that makes your clothes smell stinky. What you'll see, it kind of looks like a bunch of like little dead grass pieces everywhere. That's normal in all fungi. These individual ones are called hyphae. They're part of the fungal body. If you take all the hyphae together, that's going to be the entire fungal body. That's called the mycelium. Again, they have a cell wall. They're going to have incomplete septa, which means incomplete cell walls moving things in and out. The thing I want you to focus on is how they reproduce, because that's how we tell. Every so often in here, you get this, almost looks like a crystal ball inside of it, and then there'll be these small little tiny balls inside of it. Those little small balls are called zoospores. Think of it like a cyst, and when the cyst cracks open, these little guys can swim out in the water to go make more fungus somewhere else. They are called zoospores because they're spores, and they're zoo, animal-like, meaning they have flagella and they can swim. Okay, that gives them a unique trait. All right, so that's out of Stramenopila. Now we go to Rhodophyta, red algae. Rhodophyta, then I give you Polysiphona and Batrachiospermum. Now I'm trying to remember this from doing it in lab. So, Polysiphona. Polysiphona, when I put the picture up, if I'm trying to remember right, I gave you both generations. So if you see kind of like a Polysiphona that sticks up, but it kind of looks like a branched piece of bamboo that looks like this, that's going to be the gametophyte stage. Sorry, the sporophyte stage. That's the photosynthetic stage. So that's the sporophyte. If, however, you see near it another kind of like bamboo shoot, and coming off of it almost looks like pineapples, like fruit coming off the side of it, that's going to be the gametophyte generation. Therefore, it's going to both have male and female. So this would be N haploid, and this would be 2N diploid. All right, so again, sporophyte, gametophyte. For Batrachiospermum. Batrachiospermum, I can only find ones that showed sporophyte. But it kind of looks like that truffula tree again, where I'm going to have a small little stipe. And then I'm going to have my photosynthetic blades come out like a fuzzball. Those are the blades. And then I'm going to have another stipe, and then another fuzzball, then another stipe, and another, and it just keeps on going. But that's all going to be sporophyte for Batrachiospermum, 2N. All right, over halfway. We're in the stretch now. Now we go to the gold. Sorry, not the golden. We go to the green algae, which are then chlorophyta. All right, so chlorophyta, I gave you two examples. Both are going to be colonial. One's filamentous and one's going to colony in a ball. So Spirogyra. Spirogyra is filamentous. So now I'm going to have these algal cells linked end to end, like boxcars. It's going to have a cell wall around it. That's going to be made of cellulose. Additionally, what you will see corkscrewing through it is going to be this helical green thing. That is the chloroplast. You can also then see the nucleus in it. If you see the helical chloroplast in it, that's sporophyte. That's photosynthetic, 2N. However, what you might see, though, are some cells that do not have that helical chloroplast in it, and all you see are large balls. That would then be gametophyte. That's going to be now the reproductive stage. When you go to a volvox, volvox is a colony of balls. So it looks like think of like a big beach ball with smaller balls in the beach ball. Making the outside surface of the beach balls, you'd have all these little dots all over it. And they're basically all holding on to each other, making this orb. That is the sporophyte generation. Those are vegetative cells. They're photosynthetic. Floating on the inside of it, then, you'd have these smaller balls floating on the inside of it. Those are called daughter colonies. That's the gametophyte generation, the reproductive. Okay, both of them are chlorophyta. All right, coming on down then, we come to the last bit of slides of ones that do not have a home. Amoeba should be probably one of the ones you drew in Gen Bio. Basically looks like somebody went to a canvas with some paint and went and then splattered, what it looks like. So they're all polymorphic. There is no cell wall. So basically, just a glob, okay? You will see the nucleus. You will see the pseudopods coming out, but because these are large, blunt arms coming out, they are called lobopods. Large, blunt arms. Think of like when a white blood cell surrounds its prey in your body. Those are lobopods that come out. All right, that's all done through microfilaments. When you look at this, though, there's going to be two layers of cytoplasm. In the middle, it's going to be grainy, all in the middle. That's called the endoplasm. Right underneath the cell membrane, you're going to have a clear area, and that's called ectoplasm. It's just two areas of differentiation. All right, moving on next, then we have radiolera. Now, understand, this is Rhizopoda. This is under Sarcomastigophorid, amoeba-like creatures. Radiolerans now are the ones that live in the glass house that look like Christmas ornaments. Again, they can be any geometric shape you want. They could be cones, they could be balls. Again, with my artistic prowess, I'm sticking to a ball. So therefore, basically you have a glass ball, silicon dioxide test, and then you're going to have these large holes in it. Those are called pores. Now think, I take an amoeba-like creature and I shove it into a glass house. How can I swim? How can I feed? I take my pseudopods and I shove it against that glass house, but now I can't get these big arms coming out. What do I get? I get spikes coming out. Those spikes are called axopods, and that's how I swim, that's how I feed. All right? Now we move into another group that doesn't have a home called foraminiferan. Foraminiferan now is going to have a test, an outside shell-like area called a matrix made of calcium carbonate, chalk. Now again, they can be all shapes and sizes, like wiffle ball type shapes. The more common ones you see almost like a snail shell whorl, but there is no opening. What you would then see are little holes in it. Those are the pores. which are smaller than that of the axopods we're coming out of in radial era. Now, when I push my pseudopods against, so I push my pseudopod against it, think when you're a little kid, the Play-Doh Fun Factory, when you put the Play-Doh in, you made spaghetti, noodles came out. That's what happens now. You get these long filamentous pseudopods come out. These are called phelopods. And what do? You link them together with mucus. So as you wave them back and forth in the water, it acts like a fishing net to pick up, creatures smaller than you, algae, amoeba, whatever, and you bring them in and eat them. It also allows you to stay afloat as well, too, and swim. Last one you see there that I have for you to look at is called Physarum. Physarum is going to be from the plasmodial form. When you're a slime mold, what makes you a slime mold is you have two growth forms. You have what's called an oozing mass, which that's the technical name for it. In a lot of cases, when you look at cellular slime molds, they're the coolest things to look at because you have a petri dish. It looks like somebody went, and then you wait 5 minutes, and it starts to move on its own. It's like conscious mucus. It's fantastic to look at. However, for the plasmodials, basically what you would have is like this big, huge mass, okay? And you'd have bunch of smaller cells coming together, okay? This would be the oozing mass. Is one theory of how the birth of multicellularity could have come about from single cells to multicells. That would be the oozing mass. Somewhere in it, though, if the environment goes bad, as we said, we can go dormant. You'll see kind of like a strip that almost looks like a strip of bullets in it somewhere, where you have these large cells. And in these large cells, you'll have these balls, and those would be spores. So these creatures are identified by having an oozing mass and spore-forming bodies. All right, that ends the pre-lab for protista. Next pre-lab we'll talk about will be fungi. See ya.